My name is Wes again, Wesley Ely. I'm going to talk to you about clinical trials, but what we decided to do is to make this an interactive session where you actually get more of a hands-on, meaning questions as you wish, conversation about clinical trials rather than a didactic lecture. And Pratik and I are going to do it with you together. We figured that since both of us are here, it would be kind of a unique opportunity to take you through the story of how we developed our own clinical questions, translated those questions into clinical trials, designed and obtained funding for clinical trials, conducted the clinical trials, analyzed the data, and brought them to publication. Is that reasonable? So you're nodding your heads. I think this is, rather than go through a book chapter version of a lecture on clinical trials, this would be something more that you can apply to your life and think about what you're going to be doing in your research, and maybe we can make some trial lists out of you. I think also that people who do mixed methods work, qualitative and quantitative work, but also people who do observational cohort studies will at times learn through their observational cohort studies things that should be subjected to a clinical question in a trial. And so you might find that for the next 5 to 10 years you do something that's not a trialist work, but your research leads you down the road to develop a clinical trial. And that's the way Pratik and I have always done it. We have followed the lead. I told you yesterday, dig the break. When you find something that's unexpected or something that you didn't anticipate finding, you just merely go with the flow in your life as a researcher, as a health services researcher. And that's what Pratik and I have always done. We have had a lot of fun doing it. We now lead this center, which has well over 50 people, probably about 75 people actually in the SIB Center. And we have multiple clinical trials going. We have multiple observational cohort studies going. We have animal model work going. It's just a total gamut. We have health policy work going, clinical pharmacology work going. So we're just letting it take us wherever the questions go. But today and right now, we're going to talk about clinical trials with you. And what I'd like to do is break down the lecture, and we have about 45 minutes, lots of Q&A, lots of interaction with you. And what we'll do is we'll just take about 10 minutes to tell you the story of MENS 1 and MENS 2, which is the 2 phases of study that we've done with the alpha-2 versus the GABAergic drugs. Yesterday when you were here, I led them a little bit into that when I was telling the story of ICU delirium. But I just yesterday told you that we had pitted a major question that had not been asked in delirium world before, alpha-2 agonists versus GABAs. By the way, GABA at the VLPO and alpha-2s at the locus coeruleus. So 2 different locations in the brain. One of them hypothesized to be perhaps less deliriogenic than the other, and that was the science behind why we went with these 2 agents. And Pratik, why don't you tell a little bit about what you remember of the first development of the questions with MENS 1, and some of your recall of how we went about developing MENS 1, and then let's kind of go slow and let them ask questions along the way of what they're wondering.
Yeah, so I mean, I think it's important.
By the way, then we will go to the evolution. I didn't finish where we're going to go. After this, we're going to go to what are we doing now that we didn't used to do then. How has our clinical trial work changed in 2018 versus what it was in 22? So we will move to modern day methodology, which is quite different now than what it used to be. Okay, sorry, back to you.
So I think, one of the things that Wes taught me was to try and answer questions that have a high prevalence. And so delirium is a great area to sit and work in, and that either answer matters. And in our early work, one of the things that we were interested was delirium. We found that the rates of delirium are really high. And the next question was, are we contributing to this? Because it's great to be able to say, yes, rates are high, and if you're older, you have delirium. If you have prior cognitive impairment, you have delirium. And so Wes said, well, what's important is to try and figure out whether there is anything that is potentially modifiable. Because without that, then we can go ahead and outline all the epidemiology, but it would make no difference. And so that was one area that we were focusing on. So in our risk factor work, what that panned out was, yes, perhaps sedation and the benzodiazepines in our earlier studies being associated with delirium. And then the second part was perhaps sleep disturbances associated with delirium. And this brings to an important point with sort of doing clinical trials, is that you want to go where your strengths are, okay? So I'm an anesthesiologist, intensivist. I really don't know much about sleep medicine. And so with these two modifiable risk factors, as Wes was trying to say, well, where should we focus our attention? He is not a sleep physician, he's a pulmonologist. I'm not a sleep physician. We felt, well, let's focus where we have expertise in. So, as an anesthesiologist, sedative agents, I deal with it all the time. Let's put all our attention over there. So that was part one of why go towards sedation. You have to have your strengths over there. The second part was, as we showed that benzodiazepines were important, the next question that automatically came up was, great, you all have shown benzos are important. Now what? I mean, that's fine. You can go ahead and tell us that we shouldn't use benzos. You're giving us no alternative. And that led us to really sitting down and saying, okay, where are we going next with this? And led to two big questions. One way that we can approach this is we can modulate the sedative that we can give. So yes, if we have a benzo and we feel that GABA is something that is detrimental, we need to think of an alternative paradigm, an alpha-2. The second is, can we actually test a hypothesis where people were ridiculing us in the beginning, but fortunately J.P. Kress had just done the daily waking up trial study, is that can we actually focus on reducing or stopping sedation? So two approaches to try and hit the same problem. We knew benzos were associated with worse outcomes. Either take it away or try and come up with an alternative. And so that set off the two clinical trials that we then embarked on. And me being an anesthesiologist, I focused on sedation. Tim Gerard, Wes's second mentee at that time, pulmonary critical care intensivist, he started focusing on the daily wake up and the daily breathing trial. So that's how we divided our roles based on our own expertise, but with this common question in mind, saying, we've realized there's a problem, we realize we are contributing to the problem, and how can we actually stop that?
Okay, good. This is a good lead off. Now in business, they say you should begin with the end in mind. I think your volume will be okay with this. Begin with the end in mind. So I'm going to tell you what ends up happening with these two different ways of question asking, and then we will come back and let you ask questions about whatever you're wondering about how we design these trials. So in 2005, we began to ask the hypotheses, design clinical trials that would address the hypotheses of either an alternative receptor in the brain by which to sedate human beings in the ICU, or just use whatever receptor you want but give a lot less of it. And that was the ABC trial, which we eventually published in Lancet. Pratik's alpha-2 versus GABAergic drug was the MENS 1, which I told you yesterday was the first ever clinical trial in all of medicine to ever use delirium as a primary outcome in critical care. And that was published in JAMA. And the second MENS 2, of which we're about to finish, is propofol instead of benzos versus Dex. Jump 10, 13 years later, from 2005 we began this work to 2018, and for 10 years in all of delirium research in critical care, we had a very consistent message that delirium rates in the ICU patient, general medical or general surgical patient, were 70 to 75%. It was a very consistent number. If you were on a ventilator or in shock, that's the other thing. We chose to continue to compare apples to apples to apples to apples to apples. So all of our studies have the same basic idea. If you were all in the ICU and you were not on a ventilator, you were not very sick, but you were in the ICU for a drug overdose or something and you weren't very sick, we said we're not going to study them. We're going to study the people that we can make the most impact on. So all of you are either on a ventilator or in shock, and that's who we take. All the time in all of our studies, Brain 1, MENS, ABC, whatever, it's always you're either on a ventilator or BiPAP or in shock. So that's a group of people that had high mortality rates, 70 to 75% delirium. To 2018, if somebody asks me what is the rate of delirium in the ICU, now we're having a very, very consistent number of 40 to 50%. After doing ABC, which was the study to reduce sedatives by half using SATs, SPTs, and early mobility. Early mobility was added later. Now let's just call that the A2F bundle. And then after doing MENS 2 using dexmedetomidine instead of GABAergic drugs, we have reduced delirium very consistently by about 25% in critically ill patients. the patients who are on the vent or in shock. These people over here, the less sick people, they have rates of delirium of 20 to 40%, much lower. But we've lowered delirium by about 20–25%. Not just us by ourselves. I'm not claiming credit for all of that, but the world has done it together with all of our clinical trials. And people didn't believe that could be done. They didn't think that we could really radically change the amount of delirium that sick people had. But it just shows you how much of this was iatrogenic. And so we set out to do these clinical trials. We had to get money. We had to get support with the FDA and the NIH. And at the first, nobody would listen. Everybody said this is a dumb idea. It won't work. People won't comply. And we were basically just too persistent, too stubborn to stop. So we designed the first clinical trial, and since we couldn't get national funding from the FDA, from the VA or the NIH, we went where you could get the money. You know the Willie Sutton law? Willie Sutton, famous bank robber, why did you rob the banks? He said, that's where the money is. So we went where the money was, and that was industry. We first went to people who made the drugs and would give us funding, and we went to foundations. Industry, foundations, and federal. Those are your three pots of money by which to get grants. And so if we couldn't get it from federal, we were going to go industry and foundation. We got about $500,000 to start these studies. Now we're doing clinical trials that are $10 to $15 million. And that's about how much it costs to do a clinical trial in a governmental situation, $5 to $10 million for a large phase 2B or phase 3A study. But our first trials, we did it with $500,000. We were underfunded, it was hard, and it was somewhat of blood, sweat, and tears, but not all the way back to my SBT study. So what questions do you have about this? What is bugging you that you think you might not understand? And I call these black box items. You see maybe an idea, you see the result in a paper, but you don't understand what happened in the middle. Please start asking us some questions, and then we'll move on to what happened after MINS 1 to get Pratik to MINS 2. What are you wondering? Please, Katie.
How do you first show proof of concept and pilot data in something that you're getting so much pushback on the initial idea of just like we want to do something different and people are like it's not going to work? How do you say, well, we're going to try it in these 8 people, and the extent to which the RAD is involved with that kind of more, I would say, clinically based or clinically decision driven?
Right. I was just on the day before I got here, the reason I didn't come Tuesday, Donna, was that on Wednesday, on Tuesday afternoon, I was in a studio for a K award, a K for one of my mentees, Joellen Wilson, and she's studying catatonia right now. You know Joellen? Yeah, and Joellen's a wonderful person, and she's studying catatonia, and she wants to do a randomized controlled trial of benzos, and there's all there are case series data, and there's nothing there. So she's going to do a pilot study of benzos versus placebo for catatonia. And everybody in the room was really confused. They said, what's it going to be powered on? Because if you're going to do 20 patients, how are you going to see anything? And there won't be proof of concept there. So let me give you 3 stages. You could do a feasibility study with 20 patients just to test, and you're only going to promise that you're going to test literally feasibility of actually running the protocol. Oops. Feasibility of running the protocol. And you're not going to say that you're going to see a clinical outcome measure like reductions in catatonia, reductions in delirium. A second tier of feasibility would be what we did in the MIND study, not the MIND USA, which is about to come out in the New England Journal on October 22nd. Just got an email about that this morning, by the way, confirming October 22nd, the MIND USA study. This is the phase 3 of antipsychotics. But MIND, the original study, we did about 33 people per group, so about 110. So you can see 20 people, 110. Very different clinical world there. And for that one, when you get to 110, you probably have enough power to say you're going to look for a clinical outcome. Okay? But even there, watch what happened. If you look at that study in MIND, published in Critical Care Medicine, Tim Gerard, first author, no signal was seen in delirium reduction. So what we had to do when we went back to the NIH for the multi-million-dollar grant was say, they knew the result. We said, look, we did a feasibility study. Here's our MIND study. There's 110 people in this study. And we saw no signal. So what do you say? You don't want them to say, oh, there's no signal. We're not going to give you any money. They say, no, no, no. That's exactly our point. We don't know if these drugs work. The no signal could be real, or it could be that we were, as a type 2 error, and it was inadequately powered. What we really showed you was that we could get clinicians to give placebo. So we said from the beginning that all we were doing is designing a protocol, testing to see if we can get people in the protocol, and we weren't powering it on clinical outcomes. We were testing whether or not you could do a clinical trial. For example, when Swan-Ganz catheters were popularized by Dr. Swan and Ganz, never a proof was given. And then the support study looked at swans and said, oh, there might be increased injury from swans. And people said, well, let's go do a randomized controlled trial. We didn't know if that was possible, because we didn't know if doctors would be willing to manage patients in the ICU without a Swan-Ganz catheter. So the first things that were done were pilot studies, not to prove whether a swan worked or not, but to see if people would actually be willing to consent their patients into this clinical trial where they wouldn't get a swan. Next question.
So for that kind of in-between trial, how do you develop your aims still just with focusing on the feasibility, or do you mention, do you also talk about the clinical outcomes that you're going to look at that you're not expecting to be powered?
I'll let Pratik chime in, but I'll give you the first part of the answer and just see what's on his mind about this. We actually are, this is kind of a have your cake and eat it too situation. We're very, very cautious. So all of you are beginning your career. You're not going to go and have a multi-million-dollar grant right now to do a definitive outcomes trial. What you're going to be doing is pilot work in clinical trials. And what we're telling you is under-promise, over-deliver. Do not over-promise here. Under-promise, over-deliver. Okay? Meaning, just insist that you're testing your protocol and you're trying to determine feasibility of doing a more definitive study later on. Then you can say, we'll measure these clinical outcomes, and I'll tell you the reason why. We're going to measure these clinical outcomes, but we don't expect to see anything because it's underpowered. Okay? And that works in your favor, because if you see something, you will immediately use that in the next grant to say, oh, see, we only had this many, we already saw this, if we get it bigger. But if you see nothing, you're going to say, well, we didn't expect to see anything. It was underpowered.
But you're still showing that your methods for...
You have methodology to measure the outcomes, to analyze the outcomes.
Because that's also obviously a big thing, is that your methods are feasible.
Yes. Not just the protocol, but the methods of collection of data and analysis of outcomes. And you can use a REDCap database. You know how to put it in the REDCap database. Your statisticians know what they're doing with the data. If your outcome is delirium coma-free days in the ICU, if it's not a high death rate, you're going to use just delirium days instead of free days, that sort of thing. Pratik, what's on your mind about any of this?
Yeah, I think I echo Wes's sentiments on this. The other thing that every now and then it's helped is that as you measure the outcomes, you do get to see the effect size in the two groups. So if you're doing a feasibility study of clinical outcomes, you do see at least a signal. And if you are in sort of uncharted territory where you have no idea what these outcomes are going to pan out, then doing a larger study, you cannot do any power calculation if you have no data at all. So at least these help you say that, we saw this was the mean and standard deviation in this group, and this is the mean and standard deviation in this group in our feasibility study. And using those data, now we're trying to do the power calculation and sample size on the larger study. So it does help from that standpoint. But the feasibility part, I think, is really important. Very few granting agencies will let you embark on something without the feasibility part. The other part is that, there are various elements. When you do your preliminary section in your grant, what you want to look at is what are the areas that people may feel that you don't have strengths in, and those are areas that you want to try and tackle. So it's not that it's all work that you've done. Your team, your mentor, all that shows your group has done these various areas. So we are right now getting ready to do a prehabilitation, cognitive and physical prehabilitation for major surgery, and trying to see whether we can do that and improve outcomes. And we've shown various elements of it. We've shown that cognitive impairment happens after critical illness and hospitalization. That part is done. Okay, so we know that part is done. We have Nate Brummel, who's done cognitive therapy within the ICU. So we've said, okay, that part is done. So we know we can show feasibility that our group knows how to do it in the ICU. Our group knows how to do rehab outside the ICU. we've never done anything before. Can we actually get surgeons to let us have access to their patients for 2 weeks, 3 weeks, 4 weeks before? And so all our focus now is on that part. Wes has a similar thing, and in their part they said, yeah, it's great, you've not done a control, so we haven't seen how you can affect how this works in a control versus a non-control arm. So those are elements, whatever's missing, you want to try and hit that in your feasibility pilot study so that you have that entire portfolio. You can say, yep, now my new protocol goes from here to here, and I've shown you that I'm able to do each of these elements separately. And now I'm going to just put it together, but I have the team.
By the way, remind me at the very end of this session to tell you a story. I want to close with a story, kind of echoing what Pratik just said of go where your weaknesses are, meaning fix the weaknesses when you're in. I've got a story about that, but it's related to a cohort Brain 2 stories. I'll tell you at the end if somebody reminds me to tell them the Brain 2 story, okay? Now, Pratik just said that one of the advantages of doing a pilot feasibility study is that you can get your effect size estimates. Knowing that they're guesstimates. It's a total guesstimate. It's Frank Harrell, who's one of the world's greatest statisticians, helped to develop R. I showed you his picture yesterday. He always says, and he's at the top of the statistics world, he says, sample size calculations, power calculations are all smoke and mirrors. And you know it is, but you got to play the game anyway. But what if you don't have any estimate of effect size? What if you don't have a pilot study? You're trying to get money to do a pilot study, and you really don't have an effect size. What do you do then? What we do then is we take what would be a clinically relevant effect size. So let's say you know that delirium rates in a population that's never been studied with an interventional trial with a drug the delirium rates are 35%. And with this drug, you don't know what the reduction would be, but you do know that it wouldn't be clinically meaningful if the reduction in the delirium was only 5% or something. You say, well, then we're not going to go for a 5% reduction in delirium because nobody will care, but a clinician would care, it's your opinion, if it was a 10% reduction in delirium. Then you could say to the granting agency, we don't have an effect size for data, but we can say that a clinician wouldn't care unless it was 10%. So in order to get that sort of an effect size, we would have to study this many patients. That's a kind of way to back into things if you have nothing. But it's always better to have data on your estimated effect size. And I was just wondering, Richard, if you had any thoughts or comments about any of this statistically.
I have a whole hour talk on designing pilot studies tomorrow.
Okay, great.
I'm deleting slides.
Okay, don't do that.
You are killing me.
No. And don't be grumpy with me. He said he was a grumpy statistician.
I think we can hear it twice.
Yeah. This is, as my grandpa used to say, Wes, if you throw shit up against the barn enough time, some of it's going to stick. So let's do a little of that. All right, what's your next question? Next question. Anything? Go ahead, Elizabeth.
So, one of the difficulties I have is that I don't have a research assistant. And so the availability of me recruiting patients not only depends on my team's availability, but when I'm physically available to refer patients. How should I deal with that as I gather all my patients, that it takes a very long time to recruit all the patients, and I obviously have missed many patients that could have met inclusion criteria just because I was not available to do the study then, and I'm not sure if I should address that up front or how I should address it, because in other manuscripts I review, that's always kind of a big thing of how many people were eligible and then what was your sample size in there to make sure that you didn't pick and choose. Is that question?
Yeah, well, you're stabbing me right now because that is what drives me insane, is to think that all these people were missed who should have been enrolled and aren't, and we have no idea what the denominator really is, etc. So that's something that when I read clinical trials, and even in the New England JAMA or Lancet, the 3 best clinical journals around, when they have their consort diagrams and they do not have a denominator in there, it drives me up a wall. Or when they screen for 3 years and they say that they enrolled, 300 patients out of 500, I'm like, I don't believe you. I know that you're not being honest about how many people were really there. We know that. But so, I mean, in Mind USA, you're going to see, we screened, I think it was 20,000 people to enroll 1,200. And we track electronically every single person who would have met inclusion criteria. So I think the first thing to do is to set your sight on being as accurate as you can be about who actually meets entry criteria into your investigation. And then be honest. You say, look, I missed all these people because we didn't have time or resources to get them enrolled, but don't try and pretend that they weren't at Penn. I mean,
Yeah, exactly.
you're at Penn, okay? So if I read a paper from yours, I'm gonna know, I know that hospital. There's a crap load of patients there. You're not tricking me. Just give me the, and I know you got electronic medical records, so I know you can screen. I'm speaking to everybody in the room here. If you've got electronic medical record systems, I know you can screen these patients. I know you can track that electronically, and nobody's being tricked here. So just be honest and open, and if you really don't have a research nurse or coordinator to get them, then you're going to have to do the best you can. I call it babysitting a study. Babysitting a study means you can do all of the drawing on paper and all the design of it, but when it comes down to rubber hits the road on the daily basis, Monday, Tuesday, Wednesday, Thursday, Friday, and even Saturday, Sunday, we do 7 days a week screening. It also drives me crazy when people do 5 days a week screening and skip the weekends, because those are real people on the weekends, and there can be demographic differences from weekend admissions, from weekday admissions, so now you got another flaw in your investigation. People don't point that out, and they don't even tell you in their papers when they only did 5 day screening, and later on you find out that they did only do 5 day screening, and I go back and I say, why didn't you say that in the paper? So just be accurate, Elizabeth. Be accurate, be truthful and honest. If you don't have it, you don't have it, but you need to get it though. And when you go to Florida, try and get yourself a research nurse, because it makes it, we are very lucky. Last 2 days ago on Monday, people say they're going to try and get 2 patients per month in a study, whatever. I mean, on Monday, we enrolled 4 patients in our investigations. Our research nurses are scouring every single, it just takes that sort of energy to find every patient.
But let me just continue that for 1 second, Heidi. You know, the other thing is, as many of us, when we started our careers, we didn't have the luxury of having an entire team. And at that time, I think the team was developed by trying to get collaborators who were either our residents, our fellows, trying to, with our psychiatrists, trying to get their fellows. So, you know, with the study that we've done on pediatric delirium, no research coordinator, it's a 300 patient study, and it was done with nurse practitioners who are willing to give us some effort of their time, the residents from psychiatry who were willing to give, and it required a very elaborate sort of coordination of care, because everybody had different schedules, and trying to sit down and say, before we embark on this, we cannot do this with just Heidi and me. It's not going to happen, and trying to get that together. So, you know, you may have to expand your circle up front till you can hire people, and the only promise you can give people is academic currency. Okay, and so that's what you've got. You say, you're our collaborators, you will be co-authors with me on this paper, and if you don't have money to hire people, that's the only currency you've got. So I think starting from that, keep that as an option.
That's a great point. There are people walking around Penn who would give anything to be on a paper with you to start their academic career. So we promised academic currency, we talk about authorship very early on, and that's called free labor. But it's not free because you're paying them academically.
I just had a question about, kind of relates back to recruitment and screening. So how do you navigate an IRB that may not allow you to screen electronic health records because of HIPAA laws? So instead you have to rely on providers or the attending or someone to screen the patient for you and refer them to your team. Like that makes it so much more difficult to recruit patients.
We talk about this all the time. I'm going to let Pratik go because this is something we deal with regularly.
And I'm just going to add one more thing to that. For some, and some investigators in the hospital, like me, I'm a nurse, I may not have readily available access to the patient because I'm not a physician. So it adds that extra layer too.
Go ahead, Pratik.
So, I mean, I think this is a sort of complicated question, and we have recently dealt with it because we submitted a NIH grant, and the reviewers came back saying, we don't know, we think you're breaking all kinds of HIPAA laws by looking at screening these patients prior to them consenting. And it's just not possible to consent patients prior to screening. we went back to Gordon Bernard, who is head of our IRB, and we've gone through the regulations, and our IRB has ruled that preparatory work for research is within the federal regulations, and that is the way we have gone about rebutting the review process, which said, you cannot look at, so even though our research nurses are not technically caring for those patients, preparatory, the statement that the IRB has issued on our behalf for our grant is that the preparatory work for research is still part of the federal regulations, and it can be used for that. So you may want to bring that up to your IRB. Donna, sorry, do you want to add to that? Sorry. No, go ahead.
In other words, they accused us of breaking HIPAA rules by going into patients' charts, but we're saying, no, we're not breaking HIPAA rules. We're doing this research to prepare for their enrollment, and there's no other way for us to get in there to see if they qualify unless we do that. So we can send you verbiage on that if you want.
Yeah, that's great, thanks. I was just going to say, we have the same issue, and even though we've been working with the same health system on NIH studies since 2006, that this is a new issue that a lot of health systems are suddenly becoming worried about. And so we did the same thing, Penn State ruled, just like you said, that this is preparatory work, we're fine to do that. But the problem is, in many sites, you have to go through not just your university IRB, but also, whatever health system, and we work on several. So I think it is still a real issue. The other thing is common role. I think this is one of the things that they're hoping to help with, that you have a single IRB. But one thing that we did that was a little more helpful, we still weren't allowed into the electronic health record until we can send it, but we were allowed to give them parameters to someone else, like a clinical supervisor, that would then print us a bed board with eligible people. So if you, I'm just saying, if you come back.
Oh, I see, that's interesting. Yeah, that's a workaround.
So it's kind of a workaround, and we were completely right, but I would just tell you that some of these small health systems
Won't give you that permission.
they're worried about liability, they're worried about risk management, and even though IRB rules clearly state that this is preparatory work, they don't.
I'm going to repeat it because I want it to be on the show. The workaround is that if you can't get permission to enter the health record to screen, you can get the clinicians who are caring for the patients to do the screening and then give you a board of who's eligible. And I think that's a good workaround that small hospitals or difficult situations might find.
There are some systems, and we are trying to do that at Vandy as well, is that without actually going into the record, whether there are codes that you can write for searching the EHR, for saying that these are the criteria we are looking for in our inclusion criteria. So without anybody going, it generates, so Epic has that feature, it can automatically do it. If you actually sit down with someone and say that if people meet X, Y, Z criteria, and they're on propofol, and they're on this, print out the list. So nobody's going into that. That at least creates the first group, and it might even throw in exclusions. So that may be something to look into.
So one question, and you kind of addressed it with your beginning stages of research of gaining interest of others, even though you primarily can't pay them. But you are both lucky enough to be at Vanderbilt in a large medical center and have VA resources as well. If you're trying to establish those relationships when you may be at a small rural institution, even though perhaps it's a large institution academically, but it's isolated medically, how do you develop those partnerships and gain interest?
Are you at such an institution?
Well, no, so I'm at Penn State. So I have plenty of connections and resources, but even thinking about making those connections outside of Penn State, or even if you're at a small medical center or in a smaller institution, how do you think about those connections? Because there's academic partnerships.
This is very important. So let's say you're at Penn State and you want to do a study of rural Pennsylvania hospitals because you want to test the spread of an intervention that works in large ivory tower institutions but not in small hospitals, as an example. Well, those are dotting the state, and instead of picking them based on what you think is the right institution out in the community, rural community of Pennsylvania, you need to find the people who are interested to work with you. So, in other words, there are people out there at small institutions who do want academic currency, who are fascinated by the idea of improving care on a larger scale, and might even be frustrated that they're not at a Penn State. And there's no other way to tell you this than grassroots discovery of who those people are, where they are, and then you pick the institutions based on that. When we did MIND, for example, the first investigation of the antipsychotics, it was a multi-center trial, and we picked some fairly strange locations that you would not think, wow, why'd they get them? It's because we just found human beings at those institutions that didn't have research coordinators, that didn't whatever, and they were super pumped and very passionate. And even though it was an early idea, nobody was really caring about that idea at the time, and we didn't have a lot of money to pay them. We could only pay them academically, and we went with those people. So I'm just saying, there are people out there. You just have to search and find who. Do you have do you have a PharmD there? Do you have a physician there that wants? And through talks or statewide meetings or something like that, you can find people who do want to play, and you go with them, and you leave the other people behind. That's how we did the ICU Liberation Collaborative, by the way. On the same day, October 22nd this year, we're going to publish the 15,000 patients from the ABCDF bundle paper in critical care medicine. And we put a thing out from SCCM to the entire country, and we made them apply. So they had to tell us, we want to play. And I also added, you've got to have a bean counter, somebody from the administrative level who will sign off, not a clinician, but a person, somebody who will give you a compliance measurement person. And if they all sign off and the clinicians are there, and you bring a doctor and a nurse or a doctor and a pharmacist or whatever, then you can play. And we found 70 adult and 10 pediatric hospitals who wanted to do that. So we let them self-select themselves. And we rejected people who didn't have those teams or who didn't apply. So you got to find the right people. Oh, I'm sorry, Zoe, you were next.
It was just we were talking about finding the staff. So I think it's not just a problem of that you just don't have enough time to do it all by yourself. It's also, depending on the type of research that you're doing, really important to have independent raters. And I think nowadays you don't get away anymore with, you know, a single rater doing delirium assessment and doing whatever the index assessment is. And so we find that now earlier stuff that we're still feasibility and pilot work that we try to publish. You know, there was a student project or just me doing delirium assessment and then doing other, you know, doing the index assessment as well. That's for, you know, validating the test or something. Yeah, it's just really important that you have an independent rater. And I think just if, you know, even if ChemICU, for example, was regularly done in the ICU, it's more like a screening tool, I would say. And so we just recently got feedback from reviewers saying, you know, that's a real weakness of the study. I want you to do a comprehensive reference standard assessment, not just rely on one tool. And I think the other thing I just want to say, just from my own experience, I know, you know, this is based in the UK, and I'm a psychologist, so I work with geriatric medicine, I work with medics mostly, and we also just didn't have the money to generate pilot work at the time and money, and so it's just sort of, you know, just encourage you to think outside of the box. So we ended up going with MSc students, doing MSc clinical trials, neuropsychology. These people are dying to get close to patients, exactly, but they can't. So it's a nightmare to get the regulatory work and the approval sorted. But once you have that, you have people that you don't have to pay. They get a really strong MSc out of it. They're extremely motivated, and they have the time to spend days and weekends, you know, finding those patients. And actually, in our sort of clinical trial that I've been working on for the past 4 years, all the feasibility and pilot work came from me working together with a total of, I think, just about 10 different MSc students working together. So I think that's just, you know, there are ways of finding.
Zoe, these are great points, and just to add a few things to what you just said. When people say we can only do weekdays, we can't do weekends, I don't buy that either, because these students, they're busy Monday through Friday, but they're not busy on Saturday and Sunday. So you can always get a medical student or something like that who's dying to do some research project that'll help their career, and say, you'll be the Saturday, Sunday person. Like, absolutely, sign me up, and I will train you, and that's how you get your weekends. People say, I can't do it because I can't pay per. Just find the students. Elizabeth can go and say on her unit, say, look, I don't have a research coordinator, but Jan, you're so energetic, you're young in your career. Do you want, as a clinical nurse specialist in my unit, or as a triage nurse in my unit, would you work with me and we'll publish together, and I will train you specifically. It addresses your CAM-ICU thing. I'll train you, and in the paper I can say, a bedside nurse, triage nurse in our ICU was specifically trained to do the CAM-ICU, and that way we tested her reliability, so she was reproducible, whatever. And then the reviewer can't tell you, oh, you used a bedside nurse. Well, no, a specially trained nurse And that's how you get around it. That's different than saying, I'll just use whoever nurses are out there, that's weaker, but if you can go find people.
Yeah, so we noticed that most of our nursing staff do 3 12-hour shifts, but they realize that they have 4 days that they can, if they really want to develop and get to the next level in the nursing leadership, or going from RN1, 2, 3, 4, that they had 4 days they could contribute, and many of them actually gave up 2 days of those 4 days that they had off to come and do this. And so those are sort of great opportunities for people even at the cusp of requiring something to elevate them from RN3 to 4, etc. These are great projects to try and get them involved. And having that core group, like Wes said, not just, we have 20 people doing it, so you have no question of whether your data is actually reliable or not, but having a core group of 4 or 5 folks really helped some of our studies.
How much more time do we have? Okay, good. Let's now. Donna, you had your hand raised?
I was going to make just a real quick point. So on Friday, we are going to talk in depth about how to train these people. And we've also found when we have a lot of money that some graduate students will do this for independent study research credits when you don't have a lot of money. And then I think Pratik kind of mentioned this, but if there's a clinical ladder program in your institution, and again, as a journal editor, I would be very hesitant to publish things where the investigator was the rater. So that's really, even if you can say when I'm coming into my job, try to negotiate $50,000 to pay for a person that they would.
Yes.
One of the things we've noticed, and this goes back to your question, one of the things we've noticed is that just because it's a big university center, doesn't mean, so when we're trying to find collaborators for our multi-site studies, just because it's a big university center and a big name PI, doesn't mean that that's going to be a successful site. And actually, it's been the death of some of our sites is because the PI is, very famous, just has no time. And finding that community hospital where there is someone who's really passionate about it has actually worked out well. So we had a bedside nurse who came up at a national meeting and who said, I've not done much research. I'm really passionate about this. Can I be one of your sites? And we've actually had this person from Fort Worth, Texas, join our study. And as the lone person over there, she found a physician to be, because her IRB wouldn't allow a nurse to be a PI on an IRB, she found a physician champion to say, okay, you're my secure director, can you be on the IRB with me? So she got that person. She's got another of a nursing student, and it's been a great site. No prior experience with research, but folks like Brenda and all who are part of our research team really worked with them and got them going. So passionate, if PI, not necessarily a physician sometimes, or could be a physician, but actually is better than having a well-oiled machine, because sometimes that doesn't work well as well.
And of course, she will be the author on the paper.
Yes, of course.
Not necessarily this assigned physician who the
No.
boneheaded hospital made her get for the thing. I'm saying that because I saw Kim nodding.
Just as a follow-up with that, have you worked with any of the profit-based hospitals that are now developing all these new residencies? And like HCA, for example,
Sure.
is like popping up, I don't even know, like 10, 20 residencies a year. And so they have physicians there that have been in the community, haven't been a part of an academic world, but now they're in G&E and they're expected to do things. Has that been a resource anyone?
Yeah, that's something that we've worked with HCA, Kaiser Permanente, et cetera. We haven't used them as sites in our federally funded randomized controlled trials. I don't think we've had any of those sites there. But we've done other research projects with those sites. And sometimes they work out and sometimes they don't. I think if you asked me what's the best predictor of a successful site, I would say, you're going to hate hearing this one, Elizabeth, I would say an investigator who has a research coordinator. Okay, I mean it's extremely important, because without it, it's very difficult. But your research coordinator could become your clinical nurse specialist in your unit. It could become somebody else that you're not necessarily paying in dollars, but maybe academic currency. Because they just have to have somebody else. The lone ship person is not usually successful in enrolling in our studies. Would you agree with that?
Yeah, and so some of these HCA and other systems, they may not hire a research coordinator, but what we've had experiences with is that there are certain metrics that they hire people for. So there's a sepsis screener in one of the hospitals. There is somebody who is the fall champion, And so they all have jobs which may not be 100% jobs. And so in one of the discussions we've had, the sepsis screener who sits and goes through records said, oh, I probably have a couple of days a week that I can give. So those are places who can then become resources for you because they have some bandwidth on their primary job. Because those hospitals don't hire coordinators just waiting around for studies, but if they have effort, they can give it to you.
Let's do Tiago, and then we'll do 1 minute. I want to finish that story.
Just going back a little bit now, you said at the beginning that one of the challenges that you had was that people wouldn't believe you would get your doctors to do what you wanted them to do. So how did you actually get them to do?
What do you mean?
Like,
I'm not following.
That you're in the clinical trial you wanted to do, and that people in NIH would say, well, your physicians are not just going to be placebo versus.
Oh, right.
So how
so what we did was we go and do our own feasibility study, and then in the following submission of the grant, it says we enrolled 15, 20 people through this protocol.
But
right?
I mean, then all the physicians who were taking care of these patients were part of the study, and in that way you were able then for them to comply, or?
Well, we've done even things like we've gone to physicians' staff meetings and taken actual quantitative survey data, say, would you be willing to, and we publish that in the grant, and then we go and enroll in that same unit and prove that the patient gets through the protocol, and the rejection rate of consenting is very low and the acceptance rate is high. And so we just tackle it, and my wife makes fun of me because I always triangulate things. If we're finding directions and I ask somebody how to get there, then 5 steps later I ask another person how to get there, and one more. If I hear the same thing 3 times, I know I've got the right directions. If I hear 2 answers, I've got a problem. And she says, why are you asking again? I say, I'm triangulating. So we triangulate this problem.
But importantly, if you come up with a protocol and you think everyone's going to buy into that protocol, you're wrong. Okay, so the goal to get compliance, we do this dog and pony show in every ICU with different physician groups, go to every nursing, their unit board meeting, do all donuts, it's just to get buy-in. I've gone in and presented to my residents, and my residents have said, if you do this in my ICU, I won't let anyone get enrolled in your study. Like, all right, you're 20 years junior to me, you have the audacity to say this. You don't say that, you wait and say, okay, which is the part that is causing problems? And so ultimately, you take your protocol and you make it something that, you can't let go of the science based on just getting everyone's feedback, but you'll have enough feedback from there to be able to at least have a starting point to do your feasibility study. So you have to get buy-in, and it takes a fair amount of time because lots of people want to tweak your protocol just a little bit, and you just want to do the important things you want to tweak, but getting buy-in is really important.
So, Kim, do you want to do one last question?
Just one more, just a comment about getting buy-in, especially from those nursing units, because the nurses have to do the protocol very often. And sometimes at the large medical centers, they're doing multiple protocols. And so they, the amount of work that we're asking them to do, just one more study. So find out, talk to those nurse leaders on that unit and find out what is the feasibility of actually doing a new protocol.
Yeah, you don't want that bedside nurse, and we've had that happen to some of our studies, where as soon as you come in, the bedside nurse turns around to the family and says, they're coming to talk to you about the study. I hate the study. That's the death of that enrollment. You can guarantee it, So you have to make sure that something like that doesn't happen. We've all had our experiences.
I'll save my brain tooth story for some other time in today or tomorrow, because I'm going to end on a shorter version of the story. Remember when we started this, we told you that in the 1990s and early 2000s, every patient in the ICU basically was on a benzodiazepine. And we set out to ask the question, are these the right drugs for people to get sedated with in the ICU? Maybe in the OR, maybe one thing, but I'm talking about for multiple days of a very severe illness. And with just by 2 people sitting in a room, sitting there figuring out how can we answer that question in a clinical trial. It started with feasibility and moving to larger trials. And then triangulating the thing about reduction of drug versus different type of drug. And obviously other people on the planet trying to ask those same questions. Right now, there are about 30 studies of randomized controlled trials of benzo versus something else. in those randomized controlled trials, guess how many times a benzo, which used to be the universal only drug used for everybody, how many times the benzo beat out its competitor? 0 times. Not once. Benzos have not won a single time, and it was a usual care. Now, the other day, the story is this. The other day, we were doing MENS 2, and we have a rescue protocol for MENS 2. And if you're supposed to be randomized to dex versus propofol, but there's a rescue protocol if people are freaking out, and you have to go down a series of other drugs to use, since you can't use one of those 2 drugs, since they're these study drugs. And at one point in the rescue protocol, benzos are offered as the rescue drug. So we were up there with the nurse, and the actual patient's nurse, and I know this nurse, she's been up there for 4 years in our ICU, 4 years. Very experienced nurse, knows what she's doing, and she's been up there day in, day out with our patients. And it came time to use this benzo drip. And so we said to her, this is the next drug. This is what we're going to do in this patient because we can't unblind and we have to use a rescue drug. And the nurse got very nervous and started like oh my gosh, like you could tell she was uncomfortable. And we said, what's the matter? She goes, I've never used a benzo drip. Imagine that. Never used that in 4 years. So that was when I said, wow, this is changing practice. This is making such a qualitative difference in how these patients. And that's what you are going to do. You're going to go out there and you're going to ask clinical questions. You're going to say to yourself, is this question going to matter either answer? Do I have enough patients to answer this question? Study what you have a lot of, either answer matters. If it meets both of those criteria, then you're going to sit down and say, how do I answer this question? Who do I need? How do I answer it? What's my protocol? And start at the beginning. Walk before you can run. Okay? Thank you.
Can I, just one thing, for those of you who are doing clinical trials, realize that it takes a long time for it to happen and to reap the benefits of that. So don't get frustrated. So this story that Wes has told you, I mean, we started it in 2002, 2003, and it's the 2013 and 18 guidelines which have finally recommended benzodiazepine not to be used, right? So it takes 14, 15 years. The other thing that happens in the process is that when you all are doing clinical trials, your publication record takes a hit, okay? Because you all are waiting for your, so I suffered this with MENS being my first study. It took 5 years to get my RCT published, right? When you go in for your mentored research grants, etc., you're trying to put in for your case, they'll say, you don't have a strong publication record. So keep that in mind, that at least you're doing something that you're publishing on, so that you're not waiting. You don't want to say, I'm doing a big RCT, it's going to take me 5 years, that'll be when my first paper comes. Try and get those things done, because RCTs take a long time, okay? All right, I'm going to end. Thank you.